Welcome to the 2023 CGA-IGC podcast series, brought to you by the CGA-IGC Education Committee. Each episode aims to bring you an in-depth discussion related to emerging data, clinical challenges, and unique perspectives in the world of hereditary GI cancers. Before we start, we'd like to remind you that the content of this podcast is not intended to replace professional medical advice, diagnosis, or treatment, and the content reflects expert opinion at the time of recording. Keep listening to find out more about this episode's host, guest, and featured topic.
My name is Dr. Swati Patel. I'm a gastroenterologist and the director of the Hereditary GI Cancer Program at the University of Colorado Anschutz Medical Center. I'll be hosting today's podcast where we will learn about the most recent results published from the CAPP2 study group about the effect of resistant starch in cancer prevention for our Lynch syndrome patients. This paper received the Cancer Prevention Research Award for Outstanding Journal Article. I have the distinct honor of introducing our expert guest, Sir John Byrne, Professor of Clinical Genetics, Translational and Clinical Research Institute at Newcastle University. He's the chair of Newcastle Hospitals and the chief investigator of the CAPP3 study group. He's here today on behalf of the CAPP2 study group, including his trial co-leaders, Dr. Tim Bishop, a statistical geneticist, and Professor John Mathers, Professor of Human Nutrition at Newcastle University. Professor Byrne, thank you so much for being with us today. Let's dive right in. Can you tell us about the background of the CAPP2 study and why resistant starch was included?
Hi, Swati, and thanks again for having me join in this podcast. So the CAPP studies go back to the early 90s and in fact the late 80s. I was looking for the gene for APC with colleagues, and I was putting together a registry of familial adenomatous polyposis. I was visiting a family, and the mother had a panproctocolectomy. Her son had osteomas on his forehead. He was only 12. He just had a clear scope. And I realized he was a gene carrier because it's an extracolonic feature of the syndrome. And so on the way back with Pam, our nurse, I was just saying, I'm a physician by training. We should be doing something to prevent him getting polyps rather than just waiting for the surgeons to remove his colon. And we actually were working right next to one of our old coal mines, which hadn't quite closed. In fact, we went to visit them shortly after that and took a photograph of one of their canaries that they used to take down the mine with them to tell them if there was gas in the mine. And they still took the canaries in and they resuscitated them when they got out if they took ill. And it struck me that with FAP, we're like the canaries in the mine. If we could do something environmentally to change their risk of getting disease, it would have relevance to the rest of us. And of course, as it became apparent that APC was a gene that was a driver of sporadic colon cancer, that became even more good, strong idea. And so the interventions that we chose were actually originally chosen for our FAP study, which was published in Cancer Prevention Research in 2011 eventually. And we recruited originally 237 carriers of FAP around Europe. That was to do an intervention study with two interventions. One of them everyone's heard of, aspirin. And that was because in 1988, Gabriel Kuhn pointed out that people who took a lot of aspirin seemed to have less bowel cancer. But actually, the first idea was to give them resistant starch. And so that came from John Mathers, my long-term friend and professor of human nutrition, asking me to give a talk. And I presented to them about FAP and about how we were looking for interventions. And the rest of the conference or the workshop was about resistant starch. And the guy sitting in the front row, who I hadn't met before and only met that one time, was Dennis Burkitt. Now, Dennis was the very famous doctor who came back from Africa in the 70s and said people in Africa don't seem to get bowel cancer. And he'd spent his life doing operations there. And he came back with the idea that maybe high fiber diets in the rural Africa were protective. So we got to talking. They'd set up a research institute. We realized this was a randomizable intervention because you could give people ordinary cornstarch, or you could give them, at that time, we gave them potato starch, but it just looked like white powder. And so resistant starch and digestible starch were the same. And the idea biologically was that if you couldn't digest your carbohydrate in the upper GI tract, it would reach to the bowel where it would ferment and produce short-chain fatty acids like butyrate, which we know to be an anti-cancer agent and protective. So I thought, okay, well, we'll randomize our FAP patients to receive starch. And then along came the aspirin story. So let's use a factorial design. And the reason for that was that for the previous decade, I'd been one of the leaders of the MRC vitamin study, which is why you have folic acid in all of your food, because we were working on neural tube defects and showed that if you gave people folic acid supplements before they got pregnant, then their risk of having a spina bifida baby was reduced by 75%. That was led by Nick Wald. I was his enforcer, trying to get everyone to sign up their spina bifida patients. And I thought, well, that's a great design. We'll do the same thing. You get 2 for the price of 1. So you randomize people to either or both of those interventions or placebo. We immediately realized on FAP that getting adolescents to take diet supplements and tablets every day is a little bit of a challenge. But actually, worse than that, we realized we were going to have to video endoscope them in order to count their polyps. It's a nightmare. You could half the number of polyps, but how do you prove that in multiple centers? And so we'd literally just started the trial in 1993 when some DNA we'd sent to Richard Kolodner in Boston allowed them to identify the MSH2 gene in one of our big families. And we were off and running with Lynch syndrome, as we later called it. And immediately in 1993, I remember the moment in December of 1993 when I heard from Kolodner's group that the paper was out. I thought, we've got the wrong disease here. If we need to find these people, they don't get polyps, they just get cancer, they're older people, they're more sensible, and they'll take the tablets. So I then had to sell that to all of the so-called HNPCC International Collaborative Group. Henry was on side straight away, Hans Farsen a bit slower. JP Mecklin in Finland was very skeptical when I presented it in 1994. But then we were at a meeting in Johns Hopkins and I took him out jogging and I discovered that I could run faster than him. So he couldn't talk back while I explained the project to him. And by the time we got back to the hotel, he more or less agreed to sign up. And so it went on from there. And over the next several years, we got the funding, we got the people, and we started recruiting in 1999. The target was 1,000 recruits. In fact, we randomized rather more than that, but then we discovered that a lot of people were randomizing before they'd had a clear colonoscopy. So some of them had cancer already, some of them didn't turn up for their colonoscopy. And so we eventually actually did the trial with 937 people with Lynch syndrome in 43 centers in 16 countries. And in fact, the very last center to join was Cape Town, where I just returned from, hence the suntan. And they had a family in Namakwaland, which I went to visit with Raj Ramasar, where they go every 6 months to colonoscope them. It's a huge family. And we had 47 recruits from that extended family. What I didn't know when I signed up was it's a 700-kilometer drive in a straight line to the Namibian border where these people live. And that meant that our interventions, we were packing up packets of starch and aspirin, shipping them to South Africa, sending them 700 kilometers up the road. So it was actually quite a logistical challenge to do this. And we designed it, as I said, factorial, but also we'd realized from the then growing body of epidemiological data that the aspirin effect, which was obviously the most attractive, seemed to not happen for several years after you started taking the aspirin. So we designed the study for a 2-year intervention. When we realized it was going to be a struggle to get 1,000 people as quickly as we'd hoped, we modified the protocol to allow people to stay in for up to 4 years. So we had a range of people from 1 to 4 years on intervention. And we also put into the protocol that we would follow these people for 10 years and that our analysis would primarily target colon cancer, but we'd also count polyps and we would look for non-colorectal cancers, for the other Lynch cancers, because there was reason to think they might be affected too. And so we managed to get our study finished. We had our first meeting of the International Society Insight, which I was the first president for. We had that in Newcastle, and we rang the bell in 2005, which was the end of recruitment. And we then finished intervention in 2007. We then spent a year persuading the New England Journal of Medicine to publish the paper. And of course, journals, despite what they say, if you don't get a result, they don't like it. And so we had to say at the end of intervention, there was no impact on polyp count. And all of the received wisdom was that, well, then you might as well give up, because if you didn't prevent polyps, it's clearly not going to work. But we pressed on anyway. And when we got to the end of our funding, which was actually as it happened when the very first recruits reached their 10th year, we said, okay, it's 2009, let's do an interim analysis to see what's happening. And that led to the Lancet paper in 2011 saying that there's an effect. It was only significant in those who actually took the aspirin, but it gave us the funds to keep on going. And so when we reached 10 years, we said, let's look again. But now we actually had more information because I was very closely involved with our National Disease Registration Service. And we were able to get access with our existing consent to the medical records of all of the British recruits right through to the 2017 or 2018, which effectively gave us almost 20 years of follow-up data. And in Britain, cancer is effectively a notifiable disease. So that registry has about 100 staff around the country extracting from all the records any cancer cases. So it was a very complete analysis of the British recruits, which made up about probably a third of the total. And so then when we did the analysis in 2019, we found that it had fantastically worked in the aspirin group. So we had an intention to treat result, 50% reduction in those who took the aspirin. And that got an ASCO award and everyone thought that was great. And so we then said, let's look again at the resistant starch, because our previous interim analysis hadn't shown any effect on colon cancer. And to our amazement, when we looked at the three endpoints, we'd stopped counting polyps because we weren't really interested in them, but they didn't have the impact on colon cancer we expected. But there was a huge effect on all the other cancers. So we wrote the paper expecting the Lancet or New England Journal to snap it up. And they said, no, we don't want to publish that. And then we went to JAMA, and then we went to GUT. And then we eventually went to Cancer Prevention Research because we published CAPP2 there, and spoke to the editors and said, hey guys, this is a clinical trial. It's randomized, double-blind, international. The first half of it got an award from ASCO. This is just the other half. Why aren't you publishing this? So can we have some referees who actually read the paper? And it was accepted and published in September. And as you say, we got an award from them too. And the reason, of course, people turned it down was it was almost unbelievably good. First of all, it didn't hit the primary endpoint. And I think the reason for that, which you are well aware of, Swati, is that colon cancer and Lynch syndrome is a bit strange. Endoscopy doesn't prevent it. Our patients in Europe, the more colonoscopies they get, the more cancers they get. And clearly what's happening is that the body is kind of rejecting a lot of these cancers, I think, because of their high immunogenicity. But also we've now got some tangible evidence that many of them just appear from the surface without there having been a polyp beforehand. And so it may be that the common or garden colon cancers, where you get a polyp and lots of involvement of the microbiome and so on, may be more amenable to the resistant starch element than something which just appears out of nowhere. But the impact on the other cancers was quite dramatic. And so I've actually written down the numbers just to remind you, because I know you would like me to answer them. Sorry, I'm just talking. Do you want to stop me at any point and ask another question?
No, this is perfect. What a fascinating story. The evolution from starting with the FAP patient population that inspired this idea and sort of how it moved towards Lynch, and really is practice changing in Lynch. But please don't let me interrupt you. Keep going.
Okay. So people could sign up for either the starch or the aspirin or both. And so we had 937 people who actually started one of the treatments. And of those, 918 joined the resistant starch limb. Some of them were traveling and some couldn't handle it. So they were more or less equally split between placebo or non-placebo. And they basically got a little packet of white powder, which they mixed with their food. One lady said it made her fat, but we discovered she was mixing it with melted chocolate. But of course, resistant starch actually has a lower calorie content because it's harder to digest. So they were treated for 1 to 4 years, a mean of 25 months. And what we found was that there were essentially exactly the same number of colon cancers in the two groups after a mean of 10 years. But the non-colorectal cancers were dramatic. There were 27 in the treatment group and 48 in the placebo group. And that was actually apparent even at the 2-year data. It was 7 and 11 in the first analysis, but that had obviously risen over the years. Interestingly, overall, if you just forget about where the cancer came from, there were 78 versus 96 Lynch cancers in these patients. So it had a p-value of 0.06. So it was perilously close to being significant, even though the colon effect wasn't there. But the big impact was on the upper GI tract. There were fewer urothelials, 5 versus 10, fewer ovarian, 2 versus 5, and a lot fewer upper GI cancers. So one of the thoughts was that maybe we were having an impact on the immunological response, that somehow the starch had influenced the body in a way long-term that the immune response was better. But the other interesting idea that John Mathers came up with was secondary bile acids, because the upper GI figures are really quite striking. In the treatment group versus placebo, for stomach, it was 1 versus 3. Bile duct was none versus 3. Duodenum was 1 versus 7. Pancreas was 3 versus 8. Now, all of those cancers are in the sort of outflow zone of the bile duct of the ampulla of Vata. And secondary bile acids are a recognized carcinogen. And in fact, one of my very first pathology visits for FAP was a young man having his colon taken out. It was almost like an outflow tract from a sewage plant. Down the duodenum from the ampulla of Vata was just a string of polyps. So there's clearly unpleasant things come out in the bile. Now, you will vaguely remember from the lecture that I know you're at, that secondary bile acids are a product of the microbiome where they change the primary bile acids into these carcinogenic derivatives, which are then reabsorbed and come back out in the bile. And so what we thought maybe we had happened was that this prolonged period of resistant starch treatment had encouraged the growth of bacteria in the microbiome, which were less likely to produce these secondary bile acids, and that was a long-lasting impact, which became apparent over the years on the upper GI tract. It's obviously very exciting because it tells us, first of all, that environment is a big thing in Lynch, and we should pay more attention to the environmental issues like diet. But it also gave us a plausible explanation for what we were observing. But in truth, from a clinical point of view, there's lots of things in medicine that work, and we like acupuncture, we don't really quite work out how, but if they work, we'll use them. And so here is a treatment which is cheap and simple, and it works. So the actual figures, if you put that into the stats, the intention to treat was 0.54 with a significance value 0.01. So that's important. I'll come back to that in a moment. The second thing was we did a thing called the incident rate ratio. Now, most people who study clinical trials know about the hazard ratio, which is derived from cancer treatments where they use death as an endpoint. But if you're doing something that prevents cancers and it's genetic, you can get more than one cancer. So hazard ratio doesn't allow you for having a second primary, whereas incident rate ratio takes account of that. So it's the same idea, but you can count all the cancers, not just the first one. And the incident rate ratio was 0.37. That's a 63% reduction in these cancers. And the significance of that was 0.004. So it was a barn door result. What was interesting was, why did we have such trouble getting it published? Well, we first of all maybe didn't explain ourselves fully that this was the first analysis effectively of the trial. And indeed, A.J. Bansal and Robert Montgomery and Eduardo Villa wrote the commentary for the journal. And they were very complimentary, and they're great guys, and I know them. But actually, they made the same mistake. So even in the abstract, they said, this is a wonderful paper, but it was a secondary analysis. It wasn't a secondary analysis. It was an analysis of a secondary endpoint, which is totally different. This was always going to be analyzed. We weren't just looking at the data and filtering around and finding something interesting. So in fact, they made a series of errors in that commentary, which I'll just run through because they're actually quite important. The first one was it was a primary endpoint. The second one, it wasn't multiple testing. So there was only 3 endpoints, 2 interventions. This was statistically significant. It's not where you're looking at 1,000 targets and one of them is going to show up. The other thing was that they said that it might be affected by dropouts. And obviously we lost people along the way, but we had an intention to treat result. So even the people dropping out didn't stop the thing being significant. So that doesn't matter. And to some extent, underreporting, similarly, we know that Finland was run by gastroenterologists, so their capture of all the other cancers wasn't as effective as ours for the second decade. But most of the second decade was from the UK, and ours was complete. And actually, interestingly, we didn't put it in the main paper, but the effect was even bigger in the UK, where we had all the data. And so underreporting, of course, in a randomized trial makes no difference because it underreports on both sides of the trial. So on every level, they were kind of underestimating the impact of this data. And because of that, at the end, they said, so should we recommend resistant starch? Well, not yet. To which I shouted at my computer screen, it's taken 30 years. How long do you want to keep on going? Why would you not use this? We use colonoscopy and we still don't have any evidence it does any good, but we still use it. And it's quite dangerous. It is not dangerous to have a high fiber diet. We've been recommending it for years. So I actually profoundly disagree with them. I think now is the time to say to people with Lynch syndrome, have a high fiber diet. And in fact, the Cancer Breast Research picked up on our editorial that went to the journalists, and here we have their front page. And of course, green bananas are the big thing. The reason we started talking about green bananas was just to emphasize that this isn't some magical food stuff. You then get into the detail. So you get emails saying, I've tried to eat green bananas, I can't eat them. you're not supposed to eat them when they're sort of still rock hard green. It's just the ones that are still snappable. And at that stage, the resistant starch is crystalline, and so it doesn't digest properly. And then someone else said, but you don't get as much resistant starch as you were giving. Okay. So the answer to that is that we gave 30 grams of Novolose, which was made in New Jersey. It's a type of starch used in high fiber bread, which is about 20 grams of pure resistant starch. A big green banana will give you 7 or 8 grams of resistant starch if it's a snappable one, not yet too ripe. But of course, the average American diet has about 4 grams of resistant starch. And we weren't ever saying you just had to live on bananas. We did have one lady who phoned up and said, can you tell my husband to stop eating bananas? He's up to 23 a day and he's leaving the skins in bed. So anything is too much. What he was trying to say is that this doesn't require a massive change in your lifestyle. Switching away from rapidly digestible carbohydrates into seeds and the sort of stuff you get from the health food shops will increase your resistant starch. I should say, since it's an American podcast, you call it fermentable fiber. And that's to distinguish it from the lignins and so on, which just are bulking agents. So it's very easy to increase your resistant starch intake with something like a green banana for breakfast. It's great for journalists. They sort of say, will a banana a day keep the cancer doctor away? I was once doing a thing in a public meeting where I said, actually, also eating yesterday's pizza is pretty good too, because anything cold will have crystallized. And that went round the world. Journalists obviously like eating yesterday's pizza. So anything that's not heated, because if you heat starches, they gelatinize and become digestible. And in fact, people have suggested that the ancient people of the forest would get about 100 grams of resistant starch a day in their diet. So we really eat a very abnormal diet. And I think that that's another powerful argument for saying Lynch syndrome is to some extent a disease of the Western world, not just a genetic condition. I'll stop talking now, Swati, because you haven't had a word to say yet.
Well, it's been a fascinating conversation. I think some of the key points for our listeners are that there already is resistant starch in our diets, and the goal is to just increase that to 20 grams. And that can be accomplished with supplemental green bananas. And the exact dosage or amount, we can argue back and forth about the exact amount included in a banana. But as you mentioned, there are so many other things that we do for our Lynch syndrome patients. And when we talk about the balance of risk versus benefit, this to me and to I think our patients seems like a very low risk intervention that will likely be overall good for their health anyways.
Yeah.
Some of the questions that come up, given the potential hypotheses of how this works, is whether your group has started to think about additional ways to apply this. We certainly think about your original question, are familial adenomatous polyposis patients who have a significantly increased risk of duodenal neoplasia and ampullary cancer. Are there thoughts on expanding this to FAP populations?
Well, the very first thought was to go back to CAPP1. We kind of almost lost interest in CAPP1 because of CAPP2. And so we eventually got it published. It ran for a long time, but we had young people on resistant starch. And it was actually a rather better resistant starch. It was a mixture of potato starch and a thing called Hylon 7, which is a naturally occurring resistant starch made of cornstarch. And a lot of those took that for a long time. And of course, in FAP, duodenal disease is now our big problem. We probably won't have enough power to answer the question, but I am very keen to try and get back to that cohort. Inevitably, I'm running into sort of ethical questions about going back after such a long time. But we can argue that there's a story to tell them because of CAPP2. And so our GDPR allows me to contact them again. We're going to try and get back to them and see if there's any signal. Because when we published in the 2011 paper for CAPP1, we found that they obviously were taking the resistant starch because there was a significant impact on crypt cell proliferation. So their crypts were less lengthy. And it might be, of course, because the FAP colon cancer develops differently, that there could even be an effect on retained colonic segments in the FAP patients. Because what we have to remember is that Lynch patients represent only a minority of the general public, so to speak. Only about 15% of colon cancers follow that pathway at all, whereas the FAP patients are more representative. So that's one place to go. The other place to go is obviously people who do a lot of ERCP and upper GI work might want to try some experimental work, giving resistant starch and measuring secondary bile acids to sort of test that hypothesis. And that's obviously a very testable hypothesis as a biomarker of future benefit. The other thing, Swati, just to remember, is that we also did a secondary analysis, a true secondary analysis of the CAPP studies, looking at obesity. And that was published in 2015. And that was rather remarkable because the placebo group, the relative risk of colon cancer in the people who were obese was 2.75. I mean, it rose, I think, 0.7 per unit of BMI. And interestingly, the people who were on the aspirin had a less obvious change. So it may be that that's sort of pro-inflammatory effect of obesity, that the aspirin was having an impact on that. But it also brings us back to the point that diet is important, and that people who are slim and have a high fiber diet will have a dramatically different profile of disease than those who are overweight and eating the wrong foods. I think we generally become more and more aware of the importance of the microbiome and how we need to protect and preserve it. And indeed, there's even conversations about whether very frequent colonoscopies could even be damaging to the microbiome and actually causing us some of these problems. We're inadvertently disrupting a natural process. Rather, in my early generation, we were giving broad-spectrum antibiotics, completely unaware of the impact they were going to have. So I think it makes us look back at the whole dietary impact. And another obvious thing, having just come back from Africa and back to Burkitt's original observation, we don't get very many Lynch patients and Lynch families from around the world. It's still very much a kind of Western list of patients. Even in our countries, we're not very good at finding them because clearly it often doesn't run as a dominant trait, a visibly dominant trait. But I just wonder whether if we can start going back to the epidemiologists, doing more genomic studies to find people who've got mismatch repair gene defects and start correlating epidemiologically their dietary intake to their profile, I strongly suspect we're going to find that the penetrance of the disease is very different. And of course, if we go back to Aldred Wathin's original family that Henry rediscovered, colon cancer wasn't actually mentioned in the original report. It was gastric cancer and endometrial cancer. And so I think what we're looking at here is an environmental disease in people with a genetic predisposition, in a sense, rather than a genetic condition. And I think that's maybe going to be the long-term consequence of this study.
Yeah, and certainly those modifiers take us forward in understanding the variable penetrance, that it's not just a genetic disease, that it most certainly interacts with the environment and other risk factors.
And I think it's very empowering for people. You know, I think counselors should think that we can say to you, probably won't die of Lynch syndrome now that we know that you're at risk, because we can respond quickly to your tumors, we can scope you. It doesn't metastasize easily, so these tumors tend to stay put a long time, so they're easier to cure. But also you can do something. You can make sure your weight's okay, you get lots of exercise, have a high fiber diet, and you can significantly reduce your cancer risk. That gives them something to do. They're not just being passive recipients, they're being turned into patients from the age of 25, which I think is very important.
Yeah, and the seeds of science planted decades ago, we're now seeing some of this come to clinical practice, leveraging understanding the biology of Lynch to actually improve the care of our patients and decrease their cancer risk, whether it be in dietary, chemoprevention work that your group has done, then also understanding the immunology of the disease and leveraging that for things that, as you mentioned, Ajay Bansal is working on with Eduardo Villar Sanchez in terms of the vaccine trial that is starting here in the U.S. Well, thank you so much for your time. This has been really just a wonderful discussion that our listeners and our patients will greatly benefit from. I'll give you the last comment before we close out.
All I would say, as I said at the meeting in New Jersey, if you're going to do prevention work, it's very rewarding, but you must start at a young age because it does take a while. And I think we need to maybe re-examine how we fund our research, because these are the sorts of studies using low-cost interventions to prevent that really need more investment, but they do need very long-term investment. We were lucky to get this through to the end. I hope that Monica Bertagnoli, with whom I worked on the celecoxib study that she famously led, I hope she's listening at some point to this. I know that she's very interested in chemoprevention. We need to work out funding models that keep us working on this over the long term. and also to collaborate internationally. I think this is a demonstration of if you're going to work on rare disease, you've got to work together.
Absolutely. Well, thank you so much for being here with us. To our audience, please be sure to review the publication entitled Cancer Prevention with Resistant Starch in Lynch Syndrome Patients in the CAPP2 Randomized Placebo-Controlled Trial, a planned 10-year follow-up study published in Cancer Prevention Research. The citation can be found in our podcast descriptor. And as always, stay tuned for our next CGA-IGC podcast, where we will be reviewing the latest and greatest in hereditary GI cancers. Thanks for joining us.